Assalamu alaikum dear fellows and welcome to the channel Zoology Education. In this video we will discuss two fundamental tools of recombinant DNA technology. So the first one is restriction enzymes and second one is screening. Here is a brief introduction of restriction enzymes. Restriction enzymes are basically the enzymes that are proteins and they are produced within the bacteria. These enzymes have ability to cleave DNA, to cut DNA at specific sites. And these specific sites are known as restriction sites. Now, because of this ability to cut DNA strands at specific base sequences, these restriction enzymes are known as molecular scissors because they are also used in genetic engineering and recombinant DNA technology. So the question arises here is that what is the function of these enzymes within bacterial cell? So suppose this is a bacterial cell and this is the DNA of bacteria. When any of the foreign particle take entry into bacterial cell, the methylase enzyme of bacteria become activated and it convert the bacterial DNA into methylated bacterial DNA. Hereafter, restriction enzymes become activated and they destroy the unmethylated foreign DNA and convert it into fragments and eliminate it from the body. So, these enzymes basically act as a defensive system against viral bacteriophages. Over 400 such enzymes have been identified within bacteria, and in genetic engineering, these enzymes are used for manipulating DNA fragments. So when a restriction enzymes makes a cut at DNA, it can produce two type of ends. So the first one are the sticky ends and the second one are the blunt ends. So when a restriction enzymes cuts DNA in staggered manner, this is the staggered manner, it will produce sticky ends. And when an enzyme make a straight cut like this, it will produce blunt ends. In these diagrams, you can see a clear difference between sticky ends and blunt ends. Palm H1 is an enzyme that makes a staggered cut and it will produce sticky ends. Whereas EcoRB is an enzyme that will make a straight cut and it will produce blunt ends. So next is the recognition site for restriction enzyme. Restriction enzymes recognize DNA palindromes or palindromic sequences. These sequences are basically the sequences that are reading the same on both strands but in opposite direction. For example, GAATTC on one strand is similar to CTTAAG on the other strand when read in opposite directions. So next is the nomenclature of restriction enzymes. Since 1970s, many of the restriction enzymes have been discovered and characterized, and they are named after the source of bacterium following genus, species, and strain. For example, EcoR1 comes from Escherichia coli RY30. E comes from Escherichia, that is the genus of the bacteria from where the enzyme were isolated. CO correspond to coli, that is the specific species of the bacteria. R correspond to RY13, that is the specific strain of the bacteria, and 1 is the order of identification in the bacteria. So if we discuss the mechanism of cleavage of restriction enzyme, a restriction enzyme identify the specific DNA sequences and make a cut through the molecule by catalyzing the hydrolysis of the bond between two adjacent nucleotides. In this process, all restriction enzymes make two incisions. It will make two cuts, each through one sugar phosphate backbone of the DNA double helix. Types of restriction enzyme. So basically there are four types of restriction enzymes: type 1, type 2, type 3 and type 4. Type 1 restriction enzymes are the enzymes that cleave at sites remote from recognition site, and they require both ATP as well as S-adenosyl L-methionine. S-adenosyl L-methionine is basically a multifunctional protein and it has both activities. It can also perform restriction as well as methylase activity. Second one is type 2 enzymes. Type 2 enzymes cleave within or at a short specific distance from recognition site, and they require magnesium and they perform only a single function, that is restriction. So the third type of enzymes are the type 3 enzymes that cleave at site a short distance from recognition site and they require ATP but do not hydrolyze it. S-adenosyl L-methionine stimulate the reaction but it is not required. It exists just as a part of modification methylase. Type 4 enzymes target specific DNA that are the modified DNA, for example, methylated DNA, hydroxymethylated DNA. So here are the few applications of restriction enzymes. Restriction enzymes are widely used in genetic engineering, gene cloning, DNA fingerprinting, DNA mapping. They are used in gene sequencing as well. So next one is screening. Screening is also a very fundamental tool that is used in recombinant DNA technology. Screening is the process of identifying and separating high yielding microorganism species from their natural sources. For example, soil, and they exist in diverse microbial community within the soil. Screening is a very effective method and it allows discarding of many useless microorganisms on one hand. At the same time, it allows the easy detection of useful microorganisms that are present in population. Here are the types of screening. Screening can be of two types, primary screening or secondary screening. In primary screening, we focuses on the qualitative and indirect selection criteria, whereas in secondary screening, we focuses on both qualitative and quantitative criteria. We use primary screening when we are dealing with a large number of samples, whereas when we want to identify the best producer of the desired activity, we will prefer secondary screening. Screening relies on rapid tests that are adapted to solid mediums, whereas secondary screening uses more precise quantitative methods such as chromatography or spectrophotometry. So basically, the success of secondary screenings depend on the specific and sensitive primary selection criteria to avoid false positive. So if we discuss the examples of primary screening, it include hydrolysis halos, inhibition halos, and pH indicator changes, and it mainly focuses on the presence or absence of desired activity. Whereas secondary selection criteria involves quantitative techniques like measuring enzymatic activity or metabolic amount directly by HPLC or spectrophotometry. Blue-white screening. Blue-white screening is also a very fundamental tool that is used in recombinant DNA technology. Blue-white screening is a very effective method and it is used to identify recombinant bacteria. It is basically used to screen recombinant bacterial colonies from the non-recombinant bacterial colonies. It uses the activity of beta-galactosidase. Beta-galactosidase is basically an enzyme that is present within bacteria and it is involved in the breakdown of lactose into glucose and galactose. And the production of this enzyme is controlled by the expression of the LAGZ gene. So here is the basic working principle of blue-white screening. To screen recombinant DNA clones, X-gal, that is a chromatogenic substrate, is added to agar plates. So the beta-galactosidase, an enzyme that is present in bacteria, will hydrolyze the X-gal and it will form the blue pigment in non-recombinant colonies. As a result, the non-recombinant colonies will appear blue, whereas the recombinant colonies will remain white because the LAGZ gene in recombinant colonies has been uh, become disrupted and the beta galactosidase has not been produced. As a result, the colonies remain white because the X-gal, that is a chromatogenic substrate, has not been hydrolyzed. So the colonies will appear white. Visual screening relies on X-gal for color contrast. So the blue-white screening involves three important steps. First one is ligation. Ligation is basically joining the foreign DNA into multiple cloning site of the plasmid vector. Second one is transformation. Transformation is the incorporation of the plasmid vector with the foreign DNA into the competent bacteria. Third one is screening. Screening is basically the utilization of blue-white screening to pinpoint the recombinant bacterial colonies. So at the end, we will discuss the limitations of blue-white screening. Blue-white screening is basically only a screening procedure. It is not a selection technique. So this is the first limitation of blue-white screening. So the second limitation of the blue-white screening is that in some vectors, LAGZ gene is non-functional and it may not produce beta-galactosidase. Although the resulting colony will not be a recombinant, but it will appear white. So the next limitation is that only if a small sequence of any foreign DNA may be inserted into multiple cloning sites, it will change the reading frame of LAGZ gene and this may result into the false positive white colonies. Similarly, the small inserts within the reading frame of the LAGZ may produce ambiguous light blue colonies as the beta galactosidase is only a partially activated. So this is also a limitation of blue-white screening. So that's all about today's lecture. Thank you so much for watching the video and don't forget to like the video.